Okay, so I'm going to make a video that covers how the macromolecules, oh great, so my pen's not writing, how macromolecules are broken down in the digestive system and how they're absorbed. That you can't even read that, so let me try to find another stylus. Okay, so we're going to have carbohydrates, lipids, proteins, and nucleic acids. So you guys know that. So we're going to start with carbs, and we're going to start at the mouth, and we're going to work our way all the way through. So let me try to move this page up. Wonder how I can make a new screen. Okay. Usually this will just move, and it's not moving. My iPad's not cooperating today. Okay, so let's start in the mouth with carbohydrates. Okay, so in your mouth, what will happen is salivary amylase will be released from the salivary glands. Okay, what it does is it's going to start to break down the starch into simple sugars. Oh shoot, come on. Into simple sugars. Okay, now when salivary amylase gets to the stomach, it's inactivated. So it's inactivated by, inactivated by low pH of stomach. Okay, so then the food's going to get swallowed. Okay, so it gets swallowed, the bolus gets swallowed, it goes to the stomach. Okay, in the stomach, okay, there's no new enzymes to break down carbohydrates. So no new enzymes to break down carbs. Okay, now if I move this up, I'll get rid of that. Go back to the pen. Okay, we'll go here. Okay, now we're going to go to the small intestines. Okay, so the small intestines, pancreatic amylase will dump in from the pancreas, right? And it's going to start taking complex carbohydrates like starch and breaking them down into short strands of glucose. Okay, and individual glucose molecules. Okay, so pancreatic amylase. Okay, now starch digestion is completed. Oh, come on. Digestion is completed by these enzymes that are called brush border enzymes. Okay, these are found in the brush border of the small intestines. Okay, so in the mucosa of the small intestines is where they're found. Okay, so that's where we have things like maltase, lactase, dextrinase is another one. Okay, and they're just going to keep breaking down sugars until they get to single rings. Okay, now how are carbohydrates absorbed? Okay, so let me get rid of that. You go away. Okay, so now absorption. Okay, what happens is glucose, fructose, and galactose will be absorbed across the epithelium of the small intestines and into blood. Okay, so there's carbohydrates. Okay, now let's do a trickier one, which is proteins. Okay, so let's do proteins. We'll make proteins blue. Okay, so let's do proteins. Okay, so proteins are one important thing to remember is the enzymes that digest proteins are going to be released in an inactive form. Enzymes that digest proteins are released in an I'm getting frustrated with the stylus situation today. And this is supposed to say inactive form. Okay, so how do you recognize those? In order to recognize those, we're always looking for OGEN at the end of them, or they're going to start with pro. Okay, so OGen at the end or pro at the beginning. Okay, now, in the mouth, there's no digestion of proteins. So nothing happens in the mouth. When I was a little girl, I didn't like to eat meat, so I could sit with, a hamburger or something in my mouth for the whole time everybody else was eating, and it wouldn't go anywhere, right? So it didn't break down because there's no digestion of proteins in the mouth. Okay, now when we get to the stomach, pepsinogen, when we get to the stomach, I'm getting very frustrated with the iPad. When I touch it, it's moving. Wonder if this would be better to add text box. Pepsinogen, okay, is going to be, if I can make it bigger, is going to be activated by hydrochloric acid, okay, and it's going to become, pepsin. Okay, now pepsin will digest proteins into short strands of amino acids, okay? So AA is amino acids. Okay, now, so that's in the stomach. When we get to the small intestines, okay, pepsin is inactivated. So when the pH change, inactivated by the high pH of the small intestines, so SI small intestines. Okay, now, in the small intestines, though, we have some other enzymes, okay, and so they're going to come from the pancreas, okay, and they are trypsinogen, okay, chymotrypsinogen, and procarboxypeptidase. Okay, so 3 enzymes, inactive, they're being released from the pancreas. Okay, so trypsinogen, chymotrypsinogen, and procarboxypeptidase. Okay, now they're going to get dumped down the main pancreatic duct. Okay, so think about the ducts in the pancreas. Dump down the pancreatic duct. and they're still inactive. Okay, but what's going to happen is, let me try to move this up. Okay, what will happen is, let me get the, okay, what's going to happen is when they get to the small intestines, they actually get activated by this enzyme called enteropeptidase. So enteropeptidase, okay, activates trypsin. Okay, so it's going to activate trypsinogen. Oh, trypsinogen, autocorrect. Okay, and guess what trypsinogen is going to become when it's activated? It's going to become, oops, I'm looking for a symbol. It's going to become trypsin. Okay, now, trypsin activates chymotrypsinogen and procarboxypeptidase. And procarboxypeptidase, right? Yes, put that word in there for me. Okay, now, what do you think those two are going to become? Those will become chymotrypsin. No. Okay, so no more autocorrecting. They'll become chymotrypsin and carboxypeptidase. Okay, so let's look at that again. Okay, so when we get to the small intestines, okay, let's do when we get to the small intestines, okay, pepsin's inactivated. Okay, so pepsin only works in the stomach. Okay, the pancreas is going to release trypsinogen, chymotrypsinogen, and procarboxypeptidase. Those are inactive. Okay, they get dumped down the main pancreatic duct, and they go into what? Into the small intestines. Into the small intestines. Okay, now, there's an enzyme that's found in the small intestines. It's called and I spelled it wrong, peptidase. And what it does is it activates trypsinogen. Okay, so when trypsinogen is activated, it becomes trypsin. Trypsin then in turn activates chymotrypsinogen to be chymotrypsin and procarboxypeptidase to be carboxypeptidase, which they're kind of spelled wrong when I was typing them. Okay, now, trypsin and chymotrypsin these two things work together to break down proteins. Okay, so they're cutting in between amino acids. So you have these chains of amino acids. So they're just cutting wherever. Okay, carboxypeptidase, what it actually does is it cuts off the carboxyl end, which you guys don't have to memorize that. You might if you take nutrition. Removes the COOH from an amino acid. Okay, so basically what happens is we're chopping the proteins up until we get to these little strands of amino acids. Okay. Oh, there's my phone. Okay, now, the last thing that has to happen, let me start. Okay, so the final part of the breakdown of proteins is we have to take the like diamino acids or the two amino acids together, these peptides, and we need to break them apart. Okay, so there are enzymes in the brush border. Okay, now what's special about the enzymes in the brush border? So if this is the wall of the small intestines, okay, these brush border enzymes are going to be found in here. Okay, so they're found in these epithelial cells and in this region. So what happens is the food or the chyme has to touch them. It comes to the enzyme instead of the enzyme being released into the lumen. Okay, so there are two more enzymes that break these bonds down. One is called, it's pretty tricky, dipeptidase, which totally makes sense, right? Because it's breaking apart a dipeptide. And then there's another one that's called aminopeptidase. Okay, the big picture things about the proteins is the idea that the enzymes are inactive, right? So they're inactive. They have to be activated in order to digest proteins. Okay, now to be absorbed, let me get a better color. To be absorbed, okay, all that's going to happen is the amino acids are going to cross the epithelium of the small intestines, okay, and then they're going to go into the bloodstream. Okay, and then they can be used. They can be converted to glucose or they can be used to make new proteins. Okay, now I just had this like silly epiphany that I could erase all of this. So if I erase, then I can write over top of it. Although my erasing is not working super well. All right, that's not effective. All right, maybe we'll just push it up. All right, we'll just push it up. Okay, so now lipids. Okay, so lipids, to me, lipids are one of the trickier ones. Okay, so if we do lipids, and the reason that they're tricky, right, is because they're hydrophobic. Okay, so they don't want to dissolve in the water that's in the small intestines. So if we look at lipids, I have all these notes on talking from. Okay, so lipids. Okay, so in the mouth, okay, there's very little digestion. We do make this thing called lingual lipase, but it works in the stomach. Okay. Okay, so lingual lipase is released in the saliva, but it doesn't do anything in the mouth. It works in the stomach. Okay, now, in the stomach, okay, we have gastric lipase. Okay, now gastric lipase digests fat a little tiny bit. Okay, so tiny bit starts breaking it down. Tiny amount of digestion. Okay, and the lingual lipase also works in the stomach, but it doesn't break down a lot of fat. Okay, now the small intestines. Okay, now from the pancreas, we're going to have pancreatic lipase. Okay, now pancreatic lipase, what it's going to do is it's going to break these lipids down into monoglycerides, okay, and glycerol and fatty acids. Okay, now what's going to happen is when the fat enters the small intestines, okay, it's in big globs. Okay, so it's in these big kind of globs like this. Okay, and it's really hard for enzymes to work on this, to break it down. So what has to happen is that we're going to emulsify. Okay, so we have to emulsify the fat. So the way that we do that is we break up the globs, okay, using the motility. Okay, so we're going to mix using mixing, or the movement in the small intestines, and bile salts. Okay, so the bile salts are hydrophobic, or they're actually amphipathic. Okay, so they have a hydrophobic end. Okay, so phobic, and then they have a hydrophilic head, just like a phospholipid bilayer. Okay, so bile salts kind of look like this. So what happens is we take these globs of fat, we take these bile salts, and we mix them together. Okay, we mix it all up. And what ends up happening is we end up with these small globs of fat. Okay, and they're going to be surrounded by these bile salts. Okay, and the bile salts keep the small globs of fat from going together. Okay, from recombining. Okay, then the enzyme, okay, the lipase, can come in, okay, and break these down. And we'll end up with fatty acids and glycerol. Okay, so it breaks them down. These little things, these blobs like this, these things are actually called micelles. Okay, so it's a fat that's surrounded by a salt. Okay, now, what's going to happen, okay, is this fatty acids and the glycerol cannot be transported straight into the bloodstream because they're hydrophobic. Oh, look at that. That kind of makes it easy, except it didn't go away. Okay, so what's got to happen is we actually cover the triglycerides. Okay, so let's make the triglycerides yellow. Okay, so these are triglycerides. And what we do is we cover them with protein. Okay, so we cover them with protein. Okay, and these are called a chylomicron. And then the chylomicron, okay, so this is protein. Come on. The chylomicron actually gets absorbed. Can be absorbed. And where it gets absorbed is it actually enters the lymph vessels. Okay, so they're called lacteals, is what these specific lymph vessels are called. The lacteals of the small intestines. Okay, and then it gets unwrapped. Okay, and the fatty acids eventually will get dumped into the blood when the lymph vessel joins the blood. So I think the fats are the trickiest one because they're nonpolar. They won't dissolve in water. Okay, so I'm going to post this, and then I'll also try to find a video that someone else has done so you can watch something that someone's done with, like, professionally.